Good morning. I'm here because I would like to explain a little bit to you in a few words the project that led us to obtain a very nice grant from the European Community, the ERC grant, in 2010 that will allow our lab to work for the next 5 years. The project is called Neurocircuitry of Endocannabinoid Regulation of Food Intake, and it is based, as the title says, on the study of how the endocannabinoid system can modulate a relatively simple action of mice, that is eat or not eat, basically. This is, of course, integrated in a process, in a study of general regulation of food intake and energy balance in animals. But also for us, it is also a kind of excuse to study how the brain can take decisions about to do or not to do something. It is very well known that in general the endocannabinoid system, cannabinoid receptor type 1, CB1, are important orexigenic mediator in the brain. However, CB1 is able to control both other neurons. For example, can control both excitation and inhibition in the brain. And we know that the brain is based on, in very simplistic terms, on a balance, a tightly regulated balance between excitation and inhibition, and the outcome behavior is the result of this balance. Therefore, we think that studying how the endocannabinoid system can modulate excitation, inhibition, or neuromodulation in the brain can allow us to enter a little bit into the complexity of the brain, which is basically the word complexity is the characteristic of the brain that makes it a different organ from any other organ in the body. We are using genetic models to disentangle these aspects. We are using biochemical approaches, behavioral and pharmacological approaches to try to dissect really simple behaviors. For example, in all this project is based on the fact that hungry animals that have been fasted, decide to eat or not to eat, and we found that CB1 can still both promote this action of taking food, or even in some cases inhibit when it is acting in inhibitory neurons, for example. So, in this sense, the approach of our lab is a typical bottom-up approach to study the complexity of the brain. We are not starting from a specific disease or a specific function, but we are starting from an important system, modulator system of the brain, that is the endocannabinoid system, to try to dissect the mechanism mediating the complexity of the brain. So in this sense, this grant, this project has been recognized as important, and so we are very happy of this, and I have to thank very much all the members of the lab for the contribution in these 4 years to this process, and I thank you very much for your attention.